So good morning to everybody. Today we discuss about the synthesis of the phospholipids. What are the phospholipids? Phospholipids are the complex or compound lipids. These are complex or compound lipids. They contain the fatty acids, nitrogen base, and alcohol along with the phosphoric acid. Don't forget, they contain the phosphoric acid addition with the fatty acids, nitrogen base, and alcohol. That means so phospholipids contain the fatty acids, phosphoric acid, nitrogen base, and alcohol. These are mainly two types, that is glycerophospholipids and sphingophospholipids. These glycerophospholipids, in glycerophospholipids, glycerol as the alcohol. Here it is a glycerol as the alcohol. In sphingophospholipids, sphingosine is the alcohol. In sphingosine, sphingophospholipid, sphingosine as the alcohol. So phospholipids are complex or compound lipids. They contain the fatty acids, nitrogen base, alcohol along with the phosphoric acids. These are two types, that is glycerophospholipids and sphingophospholipids. In glycerophospholipids, glycerol acts as alcohol. In sphingophospholipids, so sphingosine acts as the alcohol. So the most important phospholipids are, these are the types of phospholipids that are the most important. That is a phosphatidylcholine, phosphatidylethanolamine, phosphatidyinositol, phosphatidylserine, phosphatidylglycerol, and plasmogens. You can remember easily all are phosphatidyl, but name of the end name is different. That is choline, ethanolamine, inositol, serine, glycerol, and the last one is the plasmogen. So that is a phosphatidylcholine, phosphatidylethanolamine, phosphatidyinositol, phosphatidylserine, diphosphatidyl and plasmogen. These are the most important glycerophospholipids. That is altogether comes under 6 categories. See the glycerophospholipids. Phosphoric acid, lecithin, cephalins, phosphatidylinositol, phosphatidylserine, plasmogens, cardiolipins. These are all comes under the glycerophospholipids. Glycerophospholipids. See here it is the these glycerophospholipids are the major lipids that is present in the biological membranes. So they consist of the glycerol 3 phosphate. These glycerol phosphate containing the glycerol 3 phosphate. This glycerol 3 phosphate esterified with the fatty acid at the carbon position 1 and 2. At the carbon position 1 and 2. That means glycerophospholipids. These glycerophospholipids containing the glycerol 3 phosphate. This glycerol 3 phosphate when esterified with the fatty acid at the carbon 1 and carbon 2. Okay. Now see the first one, phosphoric acid. Phosphoric acid is a simple form of phospholipid. It does not occur in good concentration in tissues. This phosphoric acid is intermediate in the triacyl glycerol synthesis and phospholipids. It is an intermediate in the triacyl glycerol. Lecithin. Lecithin also known as a choline. Don't forget. Cephalin is also known as a ethanolamine. Lecithin or choline, both are same. So because of the lecithin, chemical name of the lecithin is the choline. Okay. So lecithin, this is the most important abundant group of phospholipid. Important abundant group of phospholipid. So lecithin is a type of phospholipid. This is the most abundant important phospholipid that is present in the cell membrane. Chemically, it is also known as a choline. Okay, choline. Cephalins. Cephalins are ethanolamine. Why it is called the ethanolamine cephalins? So because of the ethanolamine is a nitrogen base that is present in cephalins. Okay, phosphatidylinositol. The name itself indicates phosphatidylinositol. So inositol when attached to the phosphoric acid to give the phosphatidylinositol. So when the inositol attached to the phosphoric acid to give the inositol. So this phosphatidylinositol is the most important component of the cell membrane. Certain hormones is mediated through the phosphatidylinositol. The action of certain hormones is mediated through the phosphatidylinositol. Phosphatidyl-serine. Here it is a serine is an amino acid. When the serine is attached with the phosphoric acid, produces the phosphatidyl-serine. Plasminogens. So in plasminogens, fatty acids are presented attached with the carbon one of the glycerol by the ether linkages. Here it is in plasminogens, fatty acids are attached at the carbon one of the glycerol by ether linkages. Okay, this leads to the glycerophospholipids. Okay, this result leads to the formation of the plasminogens. Cardiolipin. This is a cardiolipin is the most important, and this is isolated from the heart muscle. So this is mainly formed by the consist of two molecules of phosphoric acid. So formed by the two molecules of the phosphoric acid is held by an additional glycerol through the phosphate group. So cardiolipin, in cardiolipin, two molecules of phosphoric acid is held by an additional glycerol through the phosphate group. It is an important component of the inner mitochondrial membrane. Cardiolipin is first isolated from the heart muscle, is an important component of the inner mitochondrial membrane. See the sphingomyelins. In sphingomyelins, sphingosine is the alcohol. This is attached by the amide linkage to the fatty acid and produce the ceramide. In sphingosine, here in sphingomyelins, sphingosine is the alcohol. The sphingosine is attached to a fatty acids by the amide linkage. When the sphingosine is attached with the fatty acids by the amide linkages, produces the ceramide. Don't forget, sphingosine, when sphingosine attached with the fatty acids by amide linkages to produce the ceramide. It is the most important constituents of the myelin sheath. Sphingomyelin is an important constituent of the myelin sheath, and it is also found in good quantity in brain and nervous tissue. So brain and nervous tissue contain the sphingomyelin that is present in the myelin sheath, constituents of the myelin. And also this ceramide is act as a second messenger. By regulating the programmed cell death. Act as a second messenger by regulating the programmed cell death. The ceramide containing the 30 carbons of fatty acids is a major component of the skin and regulate the skin water permeability. So the main function of the sphingomyelins act as a second messenger by regulating the programmed cell death, and also ceramides contain the 30 carbons fatty acids. So it's a major component of the skin and regulate the skin water permeability. Now see the functions of the phospholipids. So these phospholipids are associated with the proteins from the structural components of the membrane and regulate the membrane permeability. So these phospholipids are associated with the proteins and involved in the structural components of the membrane and regulate the membrane permeability. So phospholipids in the mitochondria maintain the conformation of the electron transport chain and components and the cellular respirations. So phospholipids are when present in the mitochondria, they maintain the conformation of the electron transport chain components. They maintain the electron transport chain components and this cellular respiration. Not only they involved in the cellular respiration and also participate in the absorption of the fatty acids from the intestine. And also involved in the absorption of the fats from the intestine. So they are essential for the synthesis of the different lipoproteins, example HDL, etc., and participate in the transport of the lipids. So involved in the participates of the absorption of the fats and involved in the synthesis of the lipoproteins and participate in the transport of the lipids and prevent the accumulation of the fats that is in the liver. Prevent the accumulation of the fats in the liver. Not only the participate in the absorption of the fats and also involved in the transport of the cholesterol and helps to the removal of the cholesterol from the body. So transport the cholesterol, remove the cholesterol from the body. They act as a surfactant. And cephalins are participate in the blood clotting. Phosphatidylinositol is a major source of the second messenger that are involved in the action of some hormones. Phospholipids inositol, don't forget, is source of second messenger and involved in the action of some hormones. So the main function is these are along with the proteins and involved in the structural components of the membrane and regulate the membrane permeability. When these are present in the mitochondrial membrane, so they conform the electron transport chain components and thus regulate the cellular respiration. Not only the cellular respiration, involved in the absorption of the fats from the intestine, involved in the synthesis of the different lipoproteins, involved in the transport the cholesterol and remove the cholesterol from the body, and involved in the remove the or prevent the accumulation of the fats in the liver, and act as a surfactant, involved in the or participate in the blood clotting. Now see the metabolism of phospholipids. So these are the steps of these are the formation. So formation of the lecithin and cephalins, synthesis of phospholipids, formation of phospholipids, synthesis of phosphatidylglycerol and cardiolipin, formation of plasminogens, synthesis of the sphingomyelins. These are the metabolism of phospholipids. So metabolism of phospholipids are involved in the formation of the lecithin, cephalins, synthesis of phosphatidylglycerol, formation of the phosphatidylinositol, synthesis of the phosphatidylglycerol and cardiolipin, formation of plasminogens, synthesis of sphingomyelins. Now see the formation of lecithin and cephalins. So here it is a phosphoric acid and 1,2-diacylglycerol. These are the main products involved in the synthesis of the phospholipids, that is a phosphoric acid and 1,2-diacylglycerol. These are the main components involved in the synthesis of the phospholipids. Okay, so when phosphoric acid is converted to 1,2-diacylglycerol, 1,2-diacylglycerol, this reaction is catalyzed by the phosphatidate phosphohydrolase. Phosphatidate phosphohydrolase. So the phosphoric acid, see here it is a phosphoric acid and 1,2-diacylglycerol. These are the main sources for the synthesis of the phospholipids. These two are intermediates of the glycerol, triacylglycerol synthesis. Phosphoric acid and 1,2-diacylglycerol. These two components are intermediates in the synthesis of the triacylglycerol. Okay, the conversion of the phosphoric acid to 1,2-diacylglycerol, this reaction is catalyzed by the phosphatidate phosphohydrolase. Phosphatidate phosphohydrolase. Okay, the first one is formation of the cephalins and ethanolamine. See here it is the choline and ethanolamine. Choline and ethanolamine. These are phosphorylated and produces the phosphocholine and phosphoethanolamine. Phosphocholine and phosphoethanolamine. That means when the choline undergoes the phosphorylation, produces the phosphocholine. If the ethanolamine undergoes the phosphorylation, produces the phosphoethanolamine. Okay, this reaction is catalyzed by the kinase, that is choline kinase. So after formation of this phosphocholine and phosphoethanolamine, these are undergoes the, here it is the, when combined with the CTP, this phosphocholine or phosphoethanolamine, these are individually when combined with the CTP, produces the CDP choline and CDP ethanolamine. Okay, this phosphocholine, if the combines with the CTP, produces the CDP choline. If the phosphoethanolamine when condensed with the CTP, produces the CDP ethanolamine. Okay, this CDP choline when combined with the 1,2-diacylglycerol produces the phosphatidylcholine. This CDP choline when combined with the 1,2-diacylglycerol produces the phosphatidylcholine. Okay, if the CDP ethanolamine when combined with the 1,2-diacylglycerol produces the phosphatidylethanolamine. This reaction is catalyzed by, see here it is, this reaction is catalyzed by the phosphocholine diacylglycerol transferase. Phosphocholine diacylglycerol transferase. So here it is the choline and ethanolamine. That means is phosphatidylcholine and phosphatidylethanolamine are the pre-existing. Here it is the choline and ethanolamine are the pre-existing phospholipids. The synthesis of the phospholipids occurs in smooth endoplasmic reticulum. Don't forget, the synthesis of the phospholipids occurs in the smooth endoplasmic reticulum. This choline and ethanolamine are the pre-existing phospholipids. Hence, these reactions are together formation of the phosphatidylcholine, phosphatidylethanolamine is known as considered as a solvase pathway. Up to here it is a solvase pathway. So when the phosphatidylcholine and phosphatidylethanolamine is formed from the choline, so this choline and ethanolamine when phosphorylated produce the phosphatidylcholine, phosphatidylethanolamine. This phosphatidylcholine when combined with the CTP produce the CDP choline. If the phosphatidylethanolamine when combined with the CTP produce the CDP ethanolamine. So these two are individually and combined with the 1,2-diacylglycerol produces the phosphatidylcholine and phosphatidylethanolamine. Okay, but here it is the phosphatidylethanolamine having the capacity convert to the phosphatidylcholine. Having the capacity phosphatidylcholine, but phosphatidylcholine does not convert to the phosphatidylethanolamine. Don't forget, phosphatidylethanolamine can be converted to the phosphatidylcholine. Okay, by the methylation. By the methylation, it is converted to the phosphatidylcholine, but phosphatidylcholine does not convert to the phosphatidylethanolamine. Okay. Now see the formation of the phosphatidylserine. See the formation of the phosphatidylserine. See it is the phosphatidylethanolamine transfer its ethanol amine to the free serine. See here it is the so phosphatidylethanolamine transfer its ethanol amine to the free serine. To the free serine produce the phosphatidylserine. Produces the phosphatidylserine. This phosphatidylserine undergoes the decarboxylation. This phosphatidylserine undergoes the decarboxylation, produce the phosphatidylethanol amine. See here it is phosphatidylethanol amine. So formation of the phosphatidylserine. This is phosphatidylethanol amine. So here it is transfer its ethanol amine group to the free serine, produces the phosphatidylserine. This phosphatidylserine is undergoes the decarboxylation, produce the phosphatidyllethanol amine. So this is continuous. Here it is a reaction. See here it is. There is a continuous going on reaction. This phosphatidylethanolamine having the capacity can be converted to the phosphatidylcholine. Okay. Now see the phosphatidylinositol. See here it is phosphatidylinositol. Here it is the CDP diacylglycerol. This CDP diacylglycerol is formed from the phosphoric acid. Okay. So that means phosphoric acid involved in the formation of the CDP diacylglycerol. This reaction is catalyzed by the CTP phosphodate cytidate transferase. This reaction is catalyzed by the CTP phosphodate cytidate transferase. So after formation of this CDP diacylglycerol involved in the formation of the phosphatidylinositol. How? See here it is the CDP diacylglycerol. When combined with the inositol, see here it is combined with the inositol, produces the phosphatidylinositol. So CDP diacylglycerol when combined with the inositol produces the phosphatidylinositol. Here it is this phosphatidylinositol contain the arachidonic acid at C2 carbon. The important point here it is the phosphatidylinositol containing the arachidonic acid at the carbon 2. So this arachidonic acid is involved in the formation of the prostaglandins. Involved in the formation of the prostaglandins. Now see the formation of the phosphatidylglycerol and cardiolipin. Phosphatidylglycerol and cardiolipin. See this already I told you this CDP diacylglycerol when combined with this inositol produce the phosphatidylinositol. If the CDP diacylglycerol when combined with the glycerol 3-phosphate produce the phosphatidylglycerol 3-phosphate. produces the phosphatidylglycerol 3-phosphate. This phosphatidylglycerol 3-phosphate undergoes the hydrolation, produces the phosphatidylglycerol. So this phosphatidylglycerol 3-phosphate undergoes the hydrolation, produces the phosphatidylglycerol. Glycerol. So this phosphatidylglycerol here it is when combined with the phosphatidylglycerol produce the cardiolipin. Produces the cardiolipin. So the initial products are here it is the intermediates, that is phosphoric acid and 1,2-diacylglycerol. These two are intermediates of the that is produced in intermediates in the triacylglycerol synthesis. So when the cephalins and ethanolamine undergoes the phosphorylation, produces the phosphor Sorry for the disturbance. So phospholipids. So once again, catabolism of the glycerophospholipids. These glycerophospholipids are hydrolyzed by phospholipases, different phospholipases. So actually, these phospholipids are act on specific bond. Act on specific bond. See what are the different types of phospholipids? That is a phospholipase A1, phospholipase A2, phospholipase B, phospholipase C, and phospholipase D. You can remember easily in alphabetical, that is A1. Sorry, A, B, C, D. Under the A, you can divide it into A1 and A2. Okay, phospholipase A1, A2, phospholipase B, phospholipase C, and phospholipase D. So you can remember easily phospholipase A, B, C, D. In A, you can divide it as A1 and A2. Okay. Phospholipids, now see what are the phospholipids, which different types of phospholipids, where it is act. Okay, so that means if the carbon 1 or carbon 2 or whatever it may be. So phospholipase A1 especially cleaves the fatty acid at the C1 position. That means C1 means it's carbon 1 position. So the main function of the phospholipase A1 is the cleaves the fatty acid at the carbon 1 position. Don't forget, cleaves the fatty acid. Okay, but phospholipase A2 hydrolyzes the fatty acid at the carbon 2. So see the difference between the hydrolysis and cleaves. Here it is the A1 cleaves the fatty acid at the carbon 1, but A2 hydrolysis. Hydrolysis means here it is adding the water molecule. Okay, hydrolysis the fatty acid. Both are fatty acids, but A1 cleaves the fatty acid at the carbon 1. But A2 hydrolyzes the fatty acid at the carbon 2 position of the phospholipid. Okay. And here it is the B, that is the phospholipase B, removes the second acyl group at the carbon 2 position. So A1 cleaves the fatty acid at the carbon 1 position. A2, here it is the hydrolyzes the fatty acid at the carbon 2 position. B, that means it's phospholipase B, removes the second acyl group at the carbon 2 position. It's a remove, don't forget, remove the second acyl group at the carbon 2 position. Okay. See here it is a phospholipase C, especially cleaves the bond between the phosphate and glycerol. So phospholipase C cleaves the bond between the phosphate and glycerol. Between the phosphate and glycerol. So what about the phospholipase D? Phospholipase D hydrolyzes and remove the nitrogen base. Hydrolyzes and remove the nitrogen base. Phospholipase D. So C, here it is cleaves the bond between the phosphate and glycerol. D, that means phospholipase D, hydrolyzes and removes the nitrogen base. See here it is the phospholipase A2 is more rich sources in the snake venom and bee venom. So snake venom and bee venom are the rich sources of the A2. So not only the rich sources of the snake venom and bee venom, and also present in the tissues and pancreatic juice, and also present in the many tissues and pancreatic juice. Okay. See here it is the C, especially this phospholipase C present in the lysosomes of hepatocytes. So A2 is present in the rich sources of the snake and bee venom, not only the present in the tissues and also present in the pancreatic juice. So present in phospholipase C is present in the lysosomes of the hepatocytes. Hepatocytes. See here it is where the phospholipids especially act. Phospholipase A2 acts on the phosphatidyl inositol. So actually already I told you in previous, before this one. So phosphatidyl inositol containing the arachidonic acid at the C2. Arachidonic acid at the C2, that means it's carbon 2. This arachidonic acid, here it is so when phospholipase A2 acted upon the phosphatidyl inositol, this leads to the liberate the arachidonic acid. This arachidonic acid is involved in the synthesis of the prostaglandins. So that means here it is the phosphatidyl inositol is a sources for the prostaglandins. That means because of the phosphodiol inositol containing the arachidonic acid at the carbon 2. So this arachidonic acid is involved in the synthesis of the prostaglandins. So different phospholipases are involved in the degradation of the phospholipids. Okay, each and individual are acted upon or cleaves the different bonds. So phospholipids, these are the different water types A, B, C, D. In A, you can divide it as A1 and A2. Okay, so here it is a phospholipase. A1 is cleaves the fatty acid at the C1, that is the carbon 1 position of the phospholipid. A2 hydrolyzes the fatty acid to the carbon position to carbon 2 position of the phospholipid. Okay, there is a difference between the A1 and A2. So both are the same. Here it is the cleaves the fatty acid at the C1, and A2 is the hydrolyzes the fatty acid at the C2 position. But B is the remove the second acyl group, where that is at the carbon 2 position. Okay, so and C here it is present in the liposomes of the hepatocytes, but it cleaves the bond between the phosphate and glycerol. Phospholipase C is present in the lysosomes of hepatocytes and cleaves the bond between the phosphate and glycerol. Phospholipase D, here it is a hydrolyze and removes the nitrogen base. So fatty acids, another one is nitrogen base, another one is cleaves the bond between is the phosphate and glycerol. So another one is that is hydrolyze the fatty acids. Okay. So this is phospholipase A2, main rich sources of the snake venom and bee venom, and also present in the pancreatic juice. So after the degradation of the products of the phospholipid, that is entered into the metabolic pool and are utilized for the various purposes. See the degradation of the sphingomyelins. See here it is the degradation of the sphingomyelins. Sphingomyelinase is the most important enzyme. So this sphingomyelinase cleaves the sphingomyelin and produce the phosphoryl choline and ceramide. Produce the phosphoryl choline and ceramide. See, this sphingomyelinase is acted upon the sphingomyelin, that means cleave the sphingomyelin, produce the phosphoryl choline and ceramide. This ceramide again can be further degraded to the sphingosine and free fatty acid. Okay, this ceramide so further degrade to produce the sphingosine and free fatty acid. So what are the abnormalities of the sphingomyelins? Say the abnormalities of the sphingomyelins, that is Niemann-Pick disease and Fabus disease. Niemann-Pick disease. This Niemann-Pick disease mainly due to the deficiency of the enzyme sphingomyelinase. So what happens this deficiency of the sphingomyelinase? So due to the deficiency of the sphingomyelinase, so leads to the accumulation of the sphingomyelins. That means sphingomyelins are accumulated where that is in the liver and spleen. This leads to the enlargement of these organs. Enlargement of these organs. So characteristics of the Niemann-Pick disease, that is severe mental retardation, death may be occurring in childhood. What is the Fabus disease? This mainly due to the deficiency of the enzyme is ceramidase. So Niemann-Pick disease is mainly due to the deficiency of the sphingomyelinase. Due to the sphingomyelinase, sphingomyelins are accumulated in the liver and spleen. This leads to the enlargement of these organs. These organs, this is liver and spleens are enlarged. Okay, in Fabus disease, mainly due to the deficiency of the enzyme ceramidase. So this disorder is characterized by the skeletal deformation, subcutaneous nodules, dermatitis, and mental retardation you can observe in Fabus disease. So Fabus disease is mainly due to the deficiency of the ceramidase. Okay, now see the glycolipids. What are the glycolipids? Glycolipids are the derivatives of the ceramide. Derivatives of the ceramide, known as the glycolipids. The most important glycolipids are cerebrosides, galactocerebrosides, and glucocerebroside. Cerebrosides, galactocerebroside, and glucocerebroside. Okay. Out of these three, this galactocerebroside is a major component of the membrane lipids in the nervous tissue. And glucocerebrosides are the intermediate in the synthesis and degradation of the complex glycolipids. Intermediate in the degradation of complex and glycolipids. See here it is the sphingosine. Formation of the here it is glucocerebrosides and galactocerebrosides. Sphingosine when acylation produces the ceramide. That means when the sphingosine condenses with the acyl group produces the ceramide. This ceramide when combined with the UDP galactose, see here it is a galactose. So actually ceramide having the capacity combined with the galactose and also glucose. So if the ceramide is when combined with the galactose, produce the galactose erythrocyte. Produce the galactose erythrocyte. Okay, after formation of this galactose erythrocyte, combined with the phosphoadenosyl phosphosulfate. PAPS means is phosphoadenosyl phosphosulfate. After condensation with the PAPS, galactose erythrocyte produce the galactose erythrocyte 3 sulfate. Galactose erythrocyte 3 sulfate. Okay. So when ceramide is combined with the UDP glucose, produces the glucose erythrocyte. If the ceramide combined with the UDP galactose, produce the galactose erythrocyte, and after the condensation with the PAPS, produce the galactose erythrocyte. If the ceramide is combined with the UDP glucose, produce the glucose erythrocyte. Okay. What are the metabolic disorders of the erythrocytes? That is a Gutsch's disease, Kramp's disease, gangliosides, and sphingolipidosis. In Gutsch's disease, mainly due to the deficiency of the enzyme beta glucosidase. Beta glucosidase. When the enzyme is deficiency is beta glucosidase, leads to the tissues are having the accumulation of the glucose erythrocytes. That means glucose erythrocytes levels are increased in the tissues and leads to the liver enlargements and spleen enlargements and causes for the osteoporosis, pigmentation of the skin, anemia, and mental retardation. So Gutsch's disease is mainly due to the deficiency in the enzyme beta glucosidase. Okay, in Kramp's disease, this is mainly due to the deficiency in the enzyme beta galactosidase. Beta galactosidase. Gutsch's disease is beta glucosidase. In Kramp's disease, enzyme beta galactosidase. Okay. In this condition, accumulation of galactose erythrocytes due to the deficiency of the enzyme beta galactosidase leads to the galactose erythrocytes are accumulated and also total absence of the myelin in the nervous tissue. So total absence of the myelin sheath in the nervous tissue is a common feature. And also several mental retardation, convulsion, blindness, and deafness you can observe in Kramp's disease. Now gangliosides. What are the gangliosides? Is a complex glycosphingolipids. So this is mostly found in the gangliosides, and they contain the one or more molecules of NANA, that means is N-acetyl-neuroaminic acid. So they contain the one or more molecules of the N-acetyl-neuroaminic acid. These are bound with the ceramide oligosaccharide. Ceramide oligosaccharides. So this leads to defect in the degradation of the gangliosides, causes the gangliosides and Tay-Sachs disease. Tay-Sachs disease. So degradation of gangliosides causes the gangliosides and Tay-Sachs disease. Next one is sphingolipidasis. Sphingolipidasis is a genetic disorder. So this is mainly due to the accumulation of any one of the sphingolipids. This means maybe glycosphingolipids are accumulated or sphingoglycomides are accumulated. Sorry, sphingoglycolipids are accumulated or sphingomyelins are accumulated. So example for the sphingolipids, that is lipid storage disease. Lipid storage disease means it's the accumulation of the complex lipids. Okay. Once again. So phospholipids are the complex or compound lipids. They are having the enzyme, fatty acid, nitrogen base, alcohol, and phosphoric acid. They are mainly divided into the glycolipids and glycerophospholipids and sphingolipids. So here it is a glycerophospholipids are phosphatidylcholine, ethanolamine, inositol, serine, diphosphotidylglycerol, and plasminogens. Okay, already you know the synthesis of the here it is the metabolism of phospholipids, formation of the phospholipids involved in the formation of the lysine involved in the synthesis of phosphatidylserine, involved in the formation of phosphatidylinositol, involved in the synthesis of phosphatidylglycerol and cardiolipid, involved in the formation of the plasminogens, and involved in the synthesis of the sphingomyelins. See here it is the main initial products are the phosphatic acid and 1,2-diacylglycerol. These are the initial products for the synthesis of the phospholipids. But here it is a choline and ethanol amine. These are the pre-existing phospholipids. So phospholipid synthesis occurs in the smooth endoplasmic reticulum. So this choline and ethanolamine undergoes the phosphorylation, produces phosphocholine and phosphoethanolamine. So when these individually combined with the CTP, that means phosphocholine, if they combine with the CTP, produces CTP choline. If combined with here it is a phosphocholine ethanolamine, when combined with the CTP, produce the CDP ethanolamine. If these two are individually when combined with the 1,2-diacylglycerol, produces the phosphodiecholine and phosphodieethanolamine. Okay, this phosphodiecholine and phosphodieethanolamine are produced. That means nothing but it is choline and cephalins. Okay, so after this reaction is nothing but is called the salt-base pathway. But here is a phosphodieethanolamine having the capacity to convert to the phosphodiecholine. But phosphodiecholine does not convert to the phosphodieethanolamine. Don't forget. Okay. Now see the second one is formation of the phosphodieserine. Phosphodieethanolamine transfer its ethanol amine to the free serine, produce the phosphodieserine. So this phosphodieserine undergoes the decarboxylation, produce the phosphodieethanolamine. Okay. Third one is phosphodieinosytol, formation of the phosphodieinosytol. So for the formation of the phosphodieinosytol, we require the CDP diacylglycerol. The sources of this phosphodieinosytol is from the phosphoric acid. So phosphoric acid is converted to CDP diacylglycerol. This reaction is catalyzed by the CTP phosphodietate diacetyl transferase. Okay. This when CDP diacylglycerol combined with the inositol produce the phosphodieinosytol. Okay. Don't forget this phosphodieinosytol having the arachidonic acid with the carbon 2 position. This arachidonic acid involved in the prostaglandin synthesis or involved in the formation of the prostaglandins. Okay. Now see the formation of the phosphodieglycerol and formation of the cardiolipin. The CDP diacylglycerol when combined with the glycerol 3-phosphate produce the phosphodieglycerol 3-phosphate. So this phosphodieglycerol 3-phosphate undergoes the hydrolation, produce the phosphodieglycerol. Okay. So this phosphodieglycerol is combined with the phosphodiediglycerol, produce the cardiolipin. Okay. Now see the synthesis of the plasminogens. So for the synthesis of the plasminogens, 1-alkylate 2-acylglycerol is combined with the CDP ethanolamine, produces the 1-alkylate 2-acylglycerol 3-phosphoethanolamine. With the 1-alkylate 2-acylglycerol when combined with the CDP choline, produces the here it is 1-alkylate 2-acylglycerol 3-phosphocholine. This 1-alkylate 2-acylglycerol 3-phosphocholine when undergoes the hydrolation, produce the 1-alkylate 2-acylglycerol 3-phosphocholine. So this 3-phosphocholine when undergoes the acylation or otherwise is nothing but is condensation with the acyl CoA, produce the 1-alkylate 2-acylglycerol 3-phosphocholine. Okay. So now the formation of the sphingomyelins. For the formation of the sphingomyelins, we require the palmitoyl CoA in serine. So here it is when combined with this palmitoyl CoA combined with the serine, undergoes the different reactions or further reactions, produces the sphingosine. So this sphingosine when combined with the acyl CoA or undergoes the acylation, produce the ceramide. This ceramide is when combined with the CDP choline, produce the sphingomyelins. See the catabolism of degradation of the glycerophospholipids already you know very well is that is different phospholipids are involved in the degradation of the phospholipids.